This is a poster presentation for a project done in collaboration between Noguchi Memorial Institute for Medical Research and Pine Biotech. My name is Ilya Brodsky, and I am working together with our team at Pine Biotech, Dr. Mohit Mazumder and Dr. Harpreet Kaur, as well as our collaborators at University of Ghana, Dr. Anita Ghansah and Dr. Benedicta Mensah. Our poster is on Plasmodium falciparum, apical membrane antigen, Rhoptry Neck Protein 2 binding, which is a critical interaction for Plasmodium falciparum merozoite invasion of human host red blood cells. Several proposed malaria vaccine candidates designed to prevent such binding and limit merozoite entry have been studied with variable success at in vitro and early clinical trial stages. By performing haplotype network analysis of genomic sequencing data from sites with variable infection rates, comparing our data with MalariaGEN repository from another region in Ghana, and mapping of genomic variants onto the protein structures, we arrive at preliminary conclusions about local variation and immunogenic epitope-affecting variants for consideration. In this poster, we share preliminary findings that suggest that such genomic data can be used to develop a local strategy for efficacious vaccine design against Plasmodium falciparum blood stage infection in Ghana and surrounding regions. AMA1 RON2 binding is a critical interaction for P. falciparum merozoite invasion of host red blood cells, which causes clinical manifestation of malaria. Several proposed malaria vaccine candidates designed to prevent such binding and limit merozoite entry have been studied with variable success at in vitro and early clinical trial stages. One key observation about vaccine efficacy based on these proteins is the genetic diversity of AMA1 in parasite populations associated with disease burden, transmission rates, and various environmental factors. Our team here analyzes AMA1 genomic data from Begoro and Cape Coast in Ghana. The two towns are 240 kilometers apart and represent parasite populations in the forest and coastal savanna ecological regions of Ghana with variable transmission rates. In this project, we wanted to understand how AMA1 protein in these populations differs from the 3D7 strain of P. falciparum that is considered to be prevalent in sub-Saharan Africa and was used for the vaccine being considered in this region. 2. Identify prevalent SNPs and specific mutation signatures for each population and compare those with data from other regions in Ghana. 3. See whether SNPs in immunodominant domains are affected by variable immune pressures by exploring B cell and T cell epitopes. And 4. Ask the question about potential gene flow between parasites in these two towns located in different ecological regions. Our analysis workflow combines processing of raw data, alignment of consensus sequences for the reference 3D7 strain, haplotype analysis to determine population structure and variability, followed by identification of haplotypes specific for each population, and combinations of SNPs in haplotypes that are also specific to these populations. The analysis was conducted using new data collected from Begoro and Cape Coast in Ghana during the peak malaria transmission seasons over a 4-year period. 150 sequences from Begoro and 181 from Cape Coast were obtained by molecular inversion probe capture and deep sequencing on the Illumina MiSeq platform. Raw sequencing reads were aligned to the P. falciparum reference genome strain 3D7 using MIP Wrangler and seqDeep. Downstream analysis of synonymous and non-synonymous variants was conducted using UGene, RScripts, and the TBioInfo platform. To determine genetic diversity within and between both populations, we performed haplotype analysis based on the full sequence of AMA1 as well as AMA1 domain 1, where the binding site with RON2 is located. To calculate a haplotype distribution, we used PEGAS and APE-R packages. Haplotype networks were generated using the Templeton, Crandall, and Sting TCS method and filtering haplotypes for frequency of occurrence. Haplotypes were further analyzed for specificity of mutations to each location and their impact on protein structure. To explore the immune pressures on population structures at both locations, we explored genomic variation and predicted B cell and T cell epitopes. B cell epitopes were predicted using the BepiPred 2.0 method and TePi-tool pipeline for computational prediction of T cell epitope candidates on the IEDB analysis resource server. The predicted peptide structures were mapped onto the AMA1 protein structure 3ZWZ for downstream analysis. Analysis of genomic variation in collected samples reveals that the AMA1 in Cape Coast and Begoro have different intrapopulation diversity and many nucleotide differences from the 3D7 laboratory strain being used as the dominant strain in P. falciparum population in sub-Saharan Africa. To understand the impact of this variation on protein coding immunodominant regions, we performed analysis of non-synonymous mutations in a critical functional domain that has the potential impact on immune evasion. First, we considered the full length of AMA1. Overall, we identified common variants in 102 positions in the 1767 nucleotide long AMA1 gene, which is about 6%. We identified several positions with higher frequency of variants, including positions 116, 523, 845, 876, 988, and 995. Next, we compared prevalence of combinations of mutations using haplotype analysis. Using the TCS method, we conducted haplotype analysis, which identified 193 total haplotypes from 331 samples from Begoro and Cape Coast. Haplotype 1 is identical to P. falciparum 3D7 reference. Haplotype 1 was found in only 6 of the samples, 2 in Begoro and 4 in Cape Coast. That is less than 2% of the full population. Mutations at 6 positions were found to be common among most of the haplotypes: G116A, T523G, T845A, G876A, C988T, T995A. 4 mutations are more prevalent in Cape Coast than Begoro, and 1 mutation is more prevalent in Begoro than Cape Coast. Then, we wanted to understand differences between parasite populations in these regions of variable transmission. We found that 70% of the haplotypes had significant variation, over 26 variants or more than 1%, and 30% had minor variation from the reference P. falciparum 3D7 strain, which is between 1 and 26 variants. Of these observed haplotypes, 62 occurred at least twice and were used to construct a network which shows haplotype frequency, distribution, and divergence into specific groups. Each haplotype corresponds to a sequence which can be found in both populations or belong to a specific population only. Most haplotypes are more prevalent in Cape Coast. Next, we filtered 42 key haplotypes out of a total of 193 haplotypes with frequency difference of at least 2 between both populations. Out of these 42, 19 haplotypes are unique for Cape Coast, 15 unique for Begoro, and 7 and 1 significantly more prevalent in Cape Coast and Begoro, respectively. Constructed network shows no significant separation between the two locations, with most prevalent haplotypes shared between populations. The AMA1 protein consists of 3 domains. Domains 1 and 2 are strongly conserved across the apicomplex of phylum, suggesting a conserved function for these domains, whereas domain 3 is divergent. On the other hand, within the Plasmodium species, polymorphisms are concentrated in domain 1 and to a lesser extent domain 2. Domain 1 contains a hydrophobic groove where binding with RON2 has to take place for parasite invasion of red blood cells. Previous literature reports that the population structure of AMA1 alleles is reflective of diversifying selection under the pressure of the host immune system. We analyzed haplotype diversity by considering only AMA1 domain 1 nucleotide positions 445 to 906. Haplotype analysis yielded 93 haplotypes, 27 haplotypes that are specific to Begoro, 30 specific to Cape Coast, and 36 shared by both populations. Among the 27 haplotypes that are reasonably different between these populations in terms of frequency, difference more than 2 between populations, had 6 haplotypes that are specific to Cape Coast and 7 that are specific to Begoro. 14 haplotypes that were shared had 10 that are more prevalent in Cape Coast with a difference of at least 2 and 4 in Begoro. For domain 1 haplotypes, we observed that mutations T523G and G876A are present in all haplotypes except haplotype 1, which is identical to the reference. Mutations at 6 positions, G116A, T523G, T845A, G876A, C988T, T995A, were found to be common among most of the haplotypes. 4 mutations, T566C, A729T, C848T, A163C, are more prevalent in Cape Coast than Begoro, and 1 mutation, G106A, is more prevalent in Begoro than Cape Coast. Next, we wanted to understand how AMA1 residue conservation varies between populations. We analyzed AMA1 conservation using the AL2CO method in UCSF Chimera by calculation of positional conservation in a protein sequence alignment based on entropy. This entropy-based conservation analysis revealed variable positions specific to Begoro with high variability in Begoro and conserved in Cape Coast. For Begoro, high variability positions can be seen in red. These regions were more variable in Cape Coast. Cape Coast high variability positions can be seen in blue. These regions were more variable in Cape Coast. We then looked at where these positions are located based on AMA1 domain 1. We found that region 5, A position 221 to 224, and 3, A positions 167 to 170, are more variable in Begoro and conserved in Cape Coast. These locations are within the hydrophobic region of AMA1 RON2 binding, which is essential for merozoite entry into the red blood cell and is a target for many considered anti-malarial drugs. Conversely, region 4, AA185 to 188, is more variable in Cape Coast, while it is conserved in Begoro. This region is likely important for binding as well, since it is a part of the loop covering the hydrophobic binding region. Next, we assessed how the haplotype variants in AMA1 might reflect immune pressures specific to each region, and how these variants might affect B cell and T cell recognition by mapping identified variants on predicted T cell and B cell epitopes. 6 variants were identified to have an effect on B cell epitopes, of which 2, L189P and S283L, were more prevalent in Cape Coast, affecting B cell epitopes. 2 mutations were unique and more specific to the Cape Coast population. Others were common polymorphic sites, prevalent in haplotypes for both populations. Interestingly, one of the longest B cell epitopes, AA163 to 212, was determined to be significantly more variable in Begoro and conserved in Cape Coast, as described previously. Variant I332N, which is common in both populations, is found in one of the predicted B cell epitopes with a blossom score of -3. Analysis of predicted T cell epitopes was filtered for score above 0.9. One variant found in the predicted epitopes is Y175D, which was common for both sites and is likely to affect T cell recognition by its impact on MHC class I molecule binding specificity, Y to D Blossom 62 score of -3. Discussion. Using AMA1 sequence data generated from MIP capture and targeted deep sequencing of 181 and 150 clinical parasite isolates from Cape Coast in the coastal savanna region and Begoro in the forest region of Ghana, we observed extensive nucleotide variation in the AMA1 genomic sequences. There were 193 haplotypes, of which 70% had over 26 variants. This high genetic diversity is known to occur in high transmission areas where infected individuals usually harbor more polyclonal infections compared to those living in low transmission areas where infections are more often monoclonal. We found that circulating AMA1 haplotypes in the Cape Coast population were more diverse in distribution and uniqueness in both the full gene and the immunodominant domain 1. Despite this high level of genetic diversity, including multiple non-synonymous nucleotide substitutions, population structure shows that there is a shared gene pool between the two sites. As indicated in previous studies, the human population is likely to facilitate gene flow among falciparum parasites. Despite the ecological and epidemiological diversity between the two sites, the human movement factor is a significant consideration with implications for the spread of any emerging vaccine-resistant parasite. Of significance is the low frequency of 3D7 AMA1 reference haplotype in the two Ghanaian sites, with implications for vaccine efficacy. Several amino acid changes in domain 1 occurred in predicted B cell epitopes and may affect both naturally acquired and vaccine-induced AMA1 immunity. In addition, T cell epitope variant Y175D, circulating in both sites, is likely to affect MHC class I binding specificity, with implications for long-term vaccine efficacy. Novel sites mapped on the three-dimensional structure of AMA1 showed that these regions were located at exposed protrusions and are unique to the two Ghanaian sites. Potentially, these mutations could be affecting the binding between the two proteins in Ghana, in contrast to literature findings that common mutations like these do not affect binding affinity. This could be due to the fact that the overall pocket properties are not changed and RON2 also has variation to adapt to this new conformation. One limitation to this observation is that we did not sequence the RON2 gene in the same sites. We are planning additional studies to explore the impact of found mutations on potential binding and restrictions to proposed drug candidates aimed at this hydrophobic site. We believe that the results highlight important considerations and may provide significant value to the design of a malaria vaccine based on this antigen, and offer insights into the efficacy of AMA1 vaccines in Ghana. Further analysis on molecular dynamics of the major circulating AMA1 variants and the RON2 variants in Ghana will shed more light on the effect of the extensive genetic diversity on the red blood cell parasite invasion and vaccine efficacy for the formulation of more effective vaccines. Importantly, our study also demonstrates the utility of in-depth bioinformatic analysis for practical public health concerns in this region.